Welcome to Learn Biology, and today we're discussing the stages of meiosis, also known as meiosis to some people. So what is this meiosis? Well, it is, in its broadest definition, a process which makes gametes. Gametes are known as sex cells, and the two types of sex cells are the sperm and the oocyte, also known as the egg. The sperm is found in the male, the oocyte is found in the female. And what is the significance of meiosis? Well, we'll use the example of a human being in this to explain the significance. Here we have a human being. Human being has 23 pairs of chromosomes. 23 pairs meaning 46 chromosomes total. And that is all human beings. Normal human beings have 46 chromosomes. So when we have a male and a female mate, each which have 46 chromosomes, if meiosis did not occur, the second generation or their offspring would have 72 chromosomes. 46 plus 46 makes 72 chromosomes. So that would mean that each generation of kids would have double the amount of chromosomes from the previous generation, which is just completely abnormal. So a process known as meiosis occurs, in which it creates specialized cells for reproduction, which have half the number of chromosomes. So what this means is that the sperm has 23 chromosomes rather than 46, and the egg has 23 chromosomes rather than the 46. So when a male and female mate, we end up having 23 plus 23, which makes 46 chromosomes, which is the right number of chromosomes in the offspring. So that is the significance of meiosis. Now meiosis is split into two stages. We have meiosis 1 and we have meiosis 2. We will begin with meiosis 1. So meiosis 1 has a stage. The first stage of meiosis 1 is prophase 1, and meiosis is split into 4 stages. We have prophase 1, we have metaphase 1, anaphase 1, and telophase 1. And before we go on, if you are not familiar with prophase, metaphase, anaphase, or any of this, please check out my video on mitosis so you can have a better understanding. If you're an advanced biology student, this shouldn't be a problem. So, prophase 1. The first thing that occurs is that the nuclear membrane disintegrates on inside the cell. So the chromosomes are just floating around the cytoplasm, and they turn into we have chromosomes, so we have chromatin turning into chromosomes. So we have, loose genetic material turning into chromosomes. Spindles and centrioles appear to guide the chromosomes throughout the cell. Then a very unique step occurs, the most unique and most important step in prophase 1, which is crossing over. What occurs is that the chromosomes in prophase 1 occur in pairs known as homologous pairs, meaning identical pairs. So any chromosome occurs in sets of 2. So here, the chromosomes get so close together that a tetrad is formed. A tetrad is the definition of these chromosomes crisscrossing. And what occurs is that when they get so close together, their genetic material transfers. So genetic material transfers between the chromosomes. This is known as crossing over, and it is important because we don't know what genetic material will transfer over, so it ensures variation, makes each organism different from the others. So then we have metaphase 1. The most important term here is middle. The chromosomes, the homologous chromosomes, travel to the middle, and one is above the equator and one is below the equator. This is an invisible line, but they occur like this. And we have something known as independent assortment. Independent meaning not dependent, meaning it's random. A random assortment of where the homologous chromosomes occur, meaning if we have this one known as chromosome 1, chromosome 2, chromosome 3, and chromosome 4, this whole order could be warped, meaning chromosome 2 could be chromosome 1 and chromosome 1 could be chromosome 2. So it is very random of where these chromosomes line up on the middle of the cell. So this is metaphase 1. Independent assortment is the key term here. Then we have anaphase 1. The key term here is apart. Chromosomes move apart. So the key term here is apart. The homologous chromosomes separate, and we have one here and one here, one here and one here. So they're on the poles of the cell. So this is anaphase 1. Then we have telophase 1, in which we are turning the chromosomes, we're turning chromosomes into chromatin, so from tight to loose. And we're going to end up with 2 haploid cells, meaning this is where when we have 46 chromosomes, we're going to split the 46 into 2 cells with 23 chromosomes. So we have 2 cells with 23 chromosomes rather than the 46. And again, this is an example of a human being, not another animal. So what we end up with is 2 haploid cells. Other things that occur is a nuclear membrane forms around each set of chromosomes. So this right here is the nuclear membrane. And that is meiosis 1. We move on to meiosis 2. And this is similar to mitosis. All the stages are similar to mitosis. All we're doing here is taking the 2 haploid cells with 23 chromosomes and making an identical copy. So at the end of meiosis 2, we're going to end up with 4 cells with 23 chromosomes each. And the stages are the same: prophase 2, metaphase 2, anaphase 2, and telophase 2. And you can look each one up, and it's in my mitosis video because the steps are exactly the same. So this is the process of meiosis for you. Thank you for watching Learn Biology, and we'll see you next time.